Okay, hello everyone, and welcome to In Conversation with RTCure. My name is Manuel de la Guía Solaz, and I'm the scientific officer for this project. I will be moderating today's event. The goal of this webinar is to introduce you to the RTCure project, to discuss some of its main results, and also to give you the chance to ask some questions to the panel. Before we get started, I would like to briefly introduce IHI and mention some housekeeping points. The IHI is an EU public-private partnership, and it funds healthcare research and innovation. It brings together a broad range of scientific fields, from pharmaceuticals and biotechnology to medical technology and big data. It funds projects that address public health needs, improve the life of patients, and boost the competitiveness of the European healthcare sector. Today, we're talking about Project RTCure, which was funded under the predecessor of IHI, which was the Innovative Medicines Initiative, IMI and IMI2, which many of you may know very well. RTCure aimed to improve methods to identify those at risk of arthritis, and as well to develop new tools to help people in the earliest stages of this disease. So be aware, we will be holding a brief discussion for about 30 minutes among the panel, and there will be a dedicated question and answer session at the end of the webinar. So you're invited to post your questions to the panelists via the Q&A button throughout the discussion, and we'll be addressing them at the end of the webinar. Now, I'd like to present our speakers. So Lars, Andy, Rowan, it's a pleasure to count with you with your presence today. Could you briefly introduce yourselves and tell us what was your role within the project? Maybe we can start with Lars. Lars, you're on mute.
Yeah, so my name is Lars Karlskog. I'm Professor of Rheumatology at Karolinska Institutet in Stockholm, Sweden. I've been the academic coordinator of this, and you saw in the introductory slide all the people who have actually done the job. So I've been very happy to coordinate this.
Thank you, Lars. Andy, you are unmuted, so maybe you can go next.
Yes, good morning and welcome everybody. My name's Andy Cope. I'm Professor of Rheumatology at King's College London in London, UK. Our role in RTCure, our centre supported aspects of the research strategy and project management. And with Rowan, we co-led work package 4, which was all about developing technologies in the laboratory for analysing patient samples, and you hear more about that from Rowan. And we also contributed to another work package, which oversaw the clinical trial activities of the consortium. So more on that later.
Thank you so much. Rowan, floor is yours.
Hi, everybody. I'm Rowan Bokut. I'm a scientist at UCB, based in the UK. As you've heard from Andy already, I was heavily involved in work package 4 of RTCure. So that work package was centered around developing technologies for monitoring the RA state. And at UCB, we also were heavily involved in some of the lab work and data generation.
Thank you. Thank you all for these introductions. We're going to start with a question for Lars. Lars, could you briefly explain what rheumatoid arthritis is, and why do we need prevention and treatment for the early stages of the disease?
So rheumatoid arthritis is the most common of the inflammatory diseases that cause arthritis and destruction and soreness of joints. It's a quite severe disease, which may lead to very severe handicap if not treated appropriately. We have had many new treatments that have improved the lives for our patients a lot. But the point is that few of these treatments lead to what we really want to accomplish, a cure. And we also are often too late, meaning that the disease is going on and we have destruction of joints and also comorbidities such as lung disease and cardiovascular disease coming up if you don't treat appropriately. So what we really want to do is to understand the disease and its trajectories. And if I get the first slide, please. This slide shows how the disease develops. And indeed, we know now that the immune reactions associated with disease appear quite early as a consequence of certain genes and certain environments that we can also modify these environments. Then autoimmunity appears and evolves over time and eventually causes symptoms such as pain and fatigue, which we'll come back to, and eventually inflammation and destruction of joints. And here is where we are today, despite the therapies that we described on the right bar, today, late disease detection, too late patient involvement, and no cures. And we want to change the paradigm so that we indeed can treat much earlier and eventually also go for prevention. In the face of disease, we have some symptoms and we have autoimmunity, and that we will have early detection, also very much patient contributions, and then aiming for prevention and ultimately cure. So that's why we have this rather bold name of the project called rheumatoid tolerance for cure. And a tolerance is indeed not only treating any immune reactions that may precede development of disease, but also treating only those that are particularly involved in causing the disease, which is a real challenge. And you will hear during this webinar how we have together between many academic sites and several industries and lots of patient participation have addressed these important issues. Manuel, we don't hear you.
Of course, because I'm muted. Thank you for that. Researchers have encountered many challenges blocking the early prevention and treatment of rheumatoid arthritis. So can you tell us exactly how is RTCure different? So what did you do to address those challenges in particular?
Well, what is different is that we have indeed captured a number of academic units who have worked with these things for many years, and we have then make an alliance with industry who has technologies and also therapeutic agents that we can use. And Rowan is, in this case, a representative of one of the major industries, UCP, which contribute to this thing. And we also have a very active patient group working with us. So I think that putting these things together and then having the fact that in EU, this is a European project, we have groups which have really pioneered understanding of these early phases of the rheumatoid arthritis are actually mainly, but not only, centered in Europe. And we have many of those pioneer groups with us in this particular project. So that is some of the things that make us very well suited to take on these challenges.
Thank you, Lars. Thank you for that. I would like to remind everybody, now that we're still starting the webinar, that we will have a 15-minute Q&A session at the end of the day, at the end of 45 minutes we have allocated. So if you have any questions, feel free to use the Q&A button on the bottom right of your screen. We'll be addressing the questions at the end of the webinar. So I'm turning to you, Rowan, now. RTCure has produced many interesting results that we're going to be discussing and covering today. But it also helped reshaping the scientific methods we use for monitoring the immune reactions that we see in rheumatoid arthritis. Can you tell us how did the project contribute in this field?
Yes, thank you, Manuel. So if we have the next slide, please. So one of the key outputs from RTCure was really standardizing experimental methods across multiple labs. So as we've heard already from Lars, rheumatoid arthritis is a complex disease that involves many cells of the immune system. But when we're developing tolerizing therapies, we want to be able to monitor what changes are happening to these immune cells due to treatment. And one of the ways we do that is by analyzing cells within the blood. And it's not that easy actually to reproduce data across labs. So in order for us to understand and compare reliably across labs, it's really important for us to be able to standardize methodologies. So in RTCure, we were able to do this, and we successfully got multiple labs to follow the same protocols to monitor cells within the blood using technologies such as flow cytometry and mass cytometry. And in addition to the experimental work that we do in the lab, this also generates a lot of data. We analyze lots of samples that contain millions of cells, and we're looking at lots of parameters within those cells. So we also had to generate some data analysis pipelines and algorithms to enable us to handle all of this data. And these algorithms are also now available and being used across labs, so they're all analyzing the data in the same way. So we've now got these methodologies in place that can be used not only within the RTCure consortium, but beyond as well. And if I have the next slide, please. One of the key questions we also had was whether which antigen-specific immune reactions are present in rheumatoid arthritis and how their numbers and phenotypes change. So what do we mean by this? So within an immune reaction, the immune system maybe reacts to an antigen or a protein within the body, and we want to understand what those proteins are really, because that might help us then in developing more specific tolerizing therapies. And these experiments and these assays are a little bit more complicated to harmonize across groups, but we learned a lot along the way, and we've been able to implement methodology also to identify these antigen-specific immune reactions. And they really then form the basis of us being able to generate tolerizing therapies or identify targets for intervention in the future.
Thank you, Rowan. Thank you for this great example on the project impact on the methodology front. Andy, now there's a question for you, because we mentioned RTCure generated several great results, but I would like to ask you to identify a key result of the project and let us know why do you think this is an important achievement.
Thank you, Manuel. If we could go on to the next slide. So as Lars told you, many of the members of the RTCure consortium made pivotal contributions to understanding this at-risk state. You can see the natural history from asymptomatic to full-blown RA here on this schematic. And this at-risk state is defined by the autoimmune processes that Rowan just talked about, defined by autoantibodies or ACPA, and symptoms such as pain. And we need to be able to define this at-risk state to enroll people into these prevention trials. And you can see this window of opportunity here. Now, there are several challenges for us. One is identification of these at-risk individuals. And this is different to a clinical trial of therapy that targets established RA, where there are pre-existing cohorts of patients attending our clinics. For recruitment to this study, it was opportunistic. Family doctors referring patients in with joint pain to early arthritis clinics, and we're seeing these people for the first time. Another feature of the prevention trial, and in this particular case, the study I'm going to describe for you called APIPRA, we opted to use a drug already licensed for treating established RA, because this drug called abatacept targets some of these very early immune reactions which Rowan and Lars just talked about, specifically where T cells are activated and these initiate and drive the autoimmune process. Now, this drug abatacept is a T cell co-stimulation modulator, and it's injected weekly, and it suppresses these immune reactions responsible probably for driving the production of these antibodies. So when we detect these antibodies in the blood of these at-risk individuals, we know that the disease process has already started, and that's the window of opportunity. We know how the drug works. It has an acceptable safety profile. So that gives us some confidence when we're talking to potential trial participants. So we took individuals with joint pain, with these autoantibodies, but no joint swelling, and gave half of them placebo injections weekly for 12 months, and a similar size group, the drug abatacept, and then monitored for the development of RA, which we defined as swollen joints. Now, on the next slide, you'll see the highlight. And what this slide depicts is survival plots. Now, for those of you familiar with reading about cancer data, we talk about survival in terms of mortality. What we're talking about here is arthritis-free survival. And so you can see the results of those in the placebo group in red. So we saw progression to RA in those individuals as the curve slopes down, and also the active drug abatacept shown in blue. And you can see that those receiving the active drug for the first 12 months, the curve is almost flat, suggesting that progression to RA is prevented during this phase, contrasting with the placebo plot. When the injections are stopped at 12 months, study participants are monitored, and you can see that the survival plots start to converge. And so that means that some of the people taking the active drug then start to get RA. But the plots don't converge completely. And I think that's telling us that there may be a subset of individuals in whom there may be sustained effects and potentially prevention. Now, if you look at the next slide and think about the impact of this intervention, the key thing is that people taking abatacept felt better. And this is telling us about the symptom burden of these at-risk individuals, which has really surprised us. So when compared to placebo, pain was reduced, people felt less tired, less anxious, they were able to function better in their daily activities and slept better, and many were able to return to work. We also saw some reduction in inflammation detected by ultrasound, which is not detected clinically, and that was also reduced. And it was safe. We didn't see any difference in the side effect profile between active drug and placebo. So these results, we think, are a major milestone towards the prevention and cure, and we're seeing therapeutic effects. People feel better, and it's having an impact in terms of preventing progression. But what we now need to do is to figure out ways to switch off these immune reactions completely. In other words, to induce immune tolerance.
Thank you, Andy. This is a great result of the project, and it's actually also a great link to the next question, which is the development of tolerizing therapies. These therapies are being investigated not only in arthritis, but also in other autoimmune diseases. So, Lars, can you share with us what RTCure discovered about features of the immune system that may cause loss of tolerance, and how we can help patients with autoimmune diseases?
So thank you. So based on the technology development that Rowan showed, and now based on the great and very impactful results that Andy shown, we have shown that indeed tolerance, indeed it's possible to stop the immune reactions that cause the disease to some extent, but not completely. And that was how patients catch up during the treatment-free period. So what we need to do is both to investigate which are the particular immune reactions that cause the disease, because we want to address exactly these immune reactions, but not all immune reactions that are addressed by abatacept. And also we want to have a sustainable change of the immunity so that we turn off the disease-causing immunity permanently, and then obtaining a more permanent prevention and ultimately cure. That is within the rheumatology tolerance for cure, because normally the immune reaction, the immunity is in control of most of us and not giving rise to autoimmunity. But we break this, what we call tolerance, in some of us, and those of us develop disease. Now, the immune system has the capacity to restore this tolerance, and that is the immunity that we want to change in a very specific manner. And in RTCure, we have indeed have trials that have addressed exactly this challenging problem, and that is on the next slide. Come from one of our academic collaborators that in John Isaacs and colleagues in Newcastle. What they did was that they used data coming from studies that Rowan described, and what are the specific immunity that cause the disease. And then they took cells that normally present these type of antigens to the immune system. And these cells can either contribute to activation of the immunity or to suppression of that particular immunity. So what John and his colleagues did was they took out immune cells with leukapheresis. They put certain substances on these cells to induce a tolerance, and then they gave back these type of cells. And what they showed was, first, that this procedure was safe and that it's acceptable to patients. And they are currently now going on and having started a therapy aimed at specifically reducing the immunity that causes RA. This is by this rather complicated but very promising technology. Next slide. But what we ultimately want to do is have more simple ways of inducing this antigen-specific tolerance, using it very early. And there are many ways of doing that that have been shown to work experimentally. But the problem has been that these animal models have not really addressed the immunity that may involved and may cause rheumatoid arthritis. So what we have done here is that we have taken the particular receptors on T cells that recognize the antigens that Rowan described, putting them into mice, and then also to take the genotypes, the genes that predict the development of arthritis in human beings, and then putting them also into mice. And then we have mouse models where we could test a number of different ways. For example, different type of nanoparticles covered with antigens, or even mRNAs giving rise to tolerance. That is that we are now working with model, and we hope to have candidates for further tolerizing therapies. And then we have the opportunity, due to the fact that we have this network and we have the RTCure collaborators continuing to work together, to do this also in human beings in due time.
Thank you, Lars. I need to just give another quick reminder for the Q&A session is coming soon. For anybody in the audience having any questions on these very interesting topics that we are discussing today. So please feel free to drop them in the chat, in the Q&A box, and we will be addressing them later on after the end of the presentation. So we're a bit delayed regarding content, so I will try to speed up a little bit. But there is an important question that I have for the panelists, which is about impact, because impact is very critical for IMI and IHI projects. We always try to have the most impact on the research community, industry, patients, and the European citizens, and many stakeholders that we have at IHI. So my question to the panel is, which aspect of the project do you think is the most impactful?
I think the question goes to Andy first.
So Manuel, I think the impact here is collective. It's been a case of working across multiple partners who've had years of experience of this, working with our industry partners, having the same agenda, and getting input from our patient research partners, who very often are the people that actually keep us on track. So they have patient focus groups, they identify where the real gaps are and the need are, and they scrutinize the data as they come through. And I think if I was to put my finger on one particular aspect, I think it's been the capacity to identify and recruit people at risk who are willing to take treatment. This takes great courage, I think. And that, alongside discussions with regulators, which Lars and others led on, to try and get some insights into what is required to license a drug for prevention. This is a major step, and it's new territory. And one of the big areas we undertook was to try and develop a roadmap for this regulatory pathway, because there's no point in doing any of these experiments if we can't deliver these medications to the clinic. So it's been an amazing experience, and we've achieved a huge amount, I think, in 6 years. And we're really rolling. I'm really optimistic about the future. You're on mute, Manuel.
Classic. So thank you for that. In the interest of time, we'll go to the next question, which is actually related to impact, because the Innovative Health Initiative has been promoting a public-private approach to support research in health in Europe since 2008. So our projects demonstrated that collaborative approaches can produce impactful results that can be of benefit for the European citizens. So my question to you is, how has this public-private partnership worked for you? How did it help to reach these results and impact we've been discussing through the day?
I can begin. I think it has been an amazing project in the sense that we have the scientists from the industry, the scientists from different academias, from different sites, and we have really worked seamless together. So often in our project group, when we have had division of work into these work packages that Andy and Rowan described before, and actually when discussing, we have almost not reminded ourselves who comes from industry, comes from academia. So it has worked in an absolutely wonderful way and created a community, and this community will survive for a long time. So that will sort of enable us to really get these type of tolerizing and preventive therapies to really be implemented in reality in due time. Rowan, maybe you want to continue about how we work together between industry and academia?
Yes, definitely. Yes, so from the industry perspective, really, the public-private partnership has been really valuable in driving research forward in RA. So when thinking about trying to make new medicines or make better medicines for people, from industry, we can't do that alone. And this public-private partnership through RTCure has really allowed us to interact with key researchers in the field and really provide a platform that enables research to move forward in the future. So it's been really valuable. And I just echo what Lars said. Really, it was really difficult to tell who was from industry and who was from academia. It was really collaborative.
Thank you for your input, because we're still in time. Andy, you're free to give any comment still if you want. I just have one final question for you before the Q&A, so feel free to step in.
Yeah, so Manuel, I think we wouldn't underestimate the generous support, both in terms of time, in terms of reagent, in terms of technology, in terms of analysis that our industry partners have given, as well as funding. And we should be reminded that we got substantial funding to roll out not just one but actually 4 studies looking, experimental medicine as well as interventional trials, and our outputs would not have occurred without the support from all of the partners, each contributing unique things.
And finally, I want to just add that we also had a very active patient research partners group. And patient research partners are those patients who are involved and have experience from having RA. And they have had lots of comments, suggestions, improvements on how we should really work together with patients and getting impact on their lives.
Thank you, Lars. Patient involvement is indeed critical in this type of project. So I have a final question before the Q&A, because all projects, even the good ones, must come to an end. All funding comes to an end. All webinars come to an end. But I want to hear from you, what does the future look like for RTCure, and how will these results we've been discussing today, how they will be sustained over the long term?
Well, as you say, it's a bit of a pity that we cannot continue and there are no prolongation opportunities for successful projects. We hope that might be the case in the future. But anyway, we have created a community, and this community continues to work together. And I think that the impactful results, not only from the APIPRA that Andy described, but also for several other trials which have addressed similar type of aspects, also been very successful. That has created an understanding that prevention of autoimmune disease, here RA, is not only a fantastic vision, but is also reality. So I think that has changed the entire thinking of our community and how to think of RA and also other autoimmune disease in the future. Andy?
So I think related to that, one of the big responsibilities is to communicate the results of our efforts and to engage with our colleagues in the clinic as well as in the academic sector to help them understand what this at-risk state looks like, both in terms of symptoms, in terms of immune reactions, in terms of imaging, so that when they see these people for the first time in the clinic, they have some framework from which to work. And so the European and the US specialist societies are working very closely together as a task force to try and understand this risk stratification process. And this will be absolutely crucial when we get to the point of the regulatory process, so that we really understand risk and benefit. And as time moves on, that there will be options for when to treat and what to treat with, both in terms of inducing immune tolerance very early and using other therapies along that whole patient journey.
And of course, this would not happen if industry is not engaged with their resources and being able to put their therapies into reality. So maybe you could comment, Rowan.
Yes, I think from the sustainability perspective, I guess we've already heard from maybe the clinic and the clinical trial perspective, but I also would like to just emphasize, although RTCure has finished as a project, those interactions and collaborations are still ongoing, and I highlighted the methodologies that we generated and designed within RTCure. Well, those exist now and are still being used within labs and can also form a bit of a backbone of implementation into clinical trials and how we might monitor immune reactions or changes to the immune system in a clinical trial setting. So all of that and all of the stuff we've learned during the project can feed into future research and future clinical trial design.
Well, a big thank you to the three of you. It's great to see your sustainability plans. It's actually happening already because the project finished during last year. So we still see that there are measures being implemented for sustaining the results and taking them forward. We'll start to get questions popping in. I will start with one that goes directed to one of the members of the panel, which is Andy. So the question for you, Andy, is what about patients that are at risk of cancer development? What would be the maximum duration for a safe preventive treatment with abatacept?
So there are a number of ways to view this. We recognize that people with chronic immune-mediated inflammatory diseases have a heightened risk of cancer. So that's obviously a worry. We also know that there are some therapies which have a very small but detectable increased risk of putting people at high risk of cancer too. One of the big advantages of targeting people before they get the disease is that this risk is substantially reduced. And so, trying to prevent the disease onset in the first place will remove substantially some of that risk. Now, that's not to say that if there are people who are at risk who come up and see us in clinic where there are specific issues that we're concerned about related to cancer, that will clearly have an impact on the treatments we use. I would add a comment specifically about abatacept. I think there's been a huge amount of interest looking at the risks of cancer for a wide range of biologics. The biggest challenge that we find is because of this increased risk of cancer in people with chronic inflammatory diseases, the risk in people with RA is increased in the first place. A very good example, these people, many of them will smoke, and that puts them at a particular risk as well. So to try and identify clear signals where a particular drug is going to put someone at increase is difficult, but surveillance using pharmacovigilance and using large registries and cohorts is a key component to that. And the last thing I would say, one of the big areas that we're looking at with great interest is the idea of fixed period dosing. So if the idea is to stop someone progressing to RA, the best way to do this is a short, sharp treatment, and then you withdraw the treatment, a little bit like cancer therapy, in fact. And this is why the methods that Lars talked about, inducing tolerance, short acting, you go in, you come out. You have a drug holiday. If symptoms come back, you can go in. And I think this sort of approach will also substantially reduce risk of comorbidities, cardiovascular, infection, cancer, many of these things. So it's really important.
I could also add, Andy, that particularly concerning abatacept, in our country and as well as in a few others, we have very big registries where we follow exactly these patients and see how we could compare individuals who have abatacept with similar type of patients not having abatacept. And to our pleasure and relief, the incidence of cancers in abatacept-treated patients more or less exactly the same as for those not treated with abatacept, which is very comforting given the fact that this is just the opposite as checkpoint blockade.
Okay, thank you for this answer. Just to avoid any confusion, just to remind the audience that they can put their questions via the Q&A button. Don't get confused with the chat button, because it's the right way to do it is through the Q&A. Otherwise, it may not reach our communications team, which kindly are helping us to collect the questions. I have one question. This one is probably general for the panel, so feel free to take it as you want. It's about ethics. The question is, did you face hard issues related to the ethical aspects of treating individuals not certain to get a disease with potent drugs? And comes with a follow-up question that says, can this be mitigated with better prediction algorithms?
I think that if you begin, Andy, I could comment more about the prediction.
I think the person who raised this really important question has actually answered the question with the second question. So risk stratification is everything. It comes down to risk benefit. And we completely accept and acknowledge the fact that there will be people who may be exposed to these preventive therapies who may never go on and develop the disease. But what I would, ask the community to consider is every day we are treating people with antihypertensives, with cholesterol lowering medication, and it may be that a proportion of those individuals will never proceed to the sort of complications we see associated with high blood pressure and hypercholesterolemia. So again, we're trying to bring this concept of prevention to an at-risk population and defining the really high risk individuals. I think is absolutely key. And again, the fixed dosing period, I think, comes in here. One last point I would make, Manuel, if many people consider this numbers needed to treat concept to prevent something. So if you look in the old literature, you need to treat 50 to 60 people with aspirin to stop them getting a stroke or a heart attack. When we compute the same sort of thing for the APIPRA study for these prevention trials, you only need to treat somewhere between 4 and 6 people to prevent one from progressing. So again, this is a new field. It's something we have to become more accustomed to. But all our governments and health sectors are telling us prevention is the way forward, and we will keep pushing the boundaries.
Yeah, and an additional important thing is that those individuals we aim to prevent, and we call it personalized prevention or precision prevention, they are not healthy. They indeed have severe pain, fatigue, which often brings them to sick leave. And as Andy showed in the second slide was that also these symptoms are much mitigated during the therapy. And that means that also those who have not developed arthritis, they would be helped with this type of therapies.
Thank you, Andy. Thank you, Lars. There is a question about regulators. So what was the interaction of the project with the regulators for future guidelines for clinical trials? Was there a strong contact? Was there any contact regarding future guidelines for clinical trials aimed at prevention of RA, of rheumatoid arthritis?
Yeah, we had a long discussion with involvement of all industry partners and many of academics as well as patients. And actually, after doing that, we produced what we call a white paper, which described stratification and think about the preferred intervention in different sections of these stratified individuals having more or less risk. And we discussed this extensively with the Medical Products Agency in Sweden, because at the time EMA office was moving to Amsterdam and was not so available. So we got a very positive response from the MPA in Sweden and got some advice. So I think that hopefully that document can serve as a basis for future discussions on how companies, when they come to EMA, how they could use these documents in order to stratify and rectify eventually in problems in ethics and in stratification. Andy?
So Manuel, in addition to the meeting with the Swedish MPA, where they reviewed this briefing document asking questions about the appropriateness of the at-risk population and the need for treatments, several of us also engaged the MHRA in London, asking very similar questions and asking specifically about the trial design, because this is where having the input of our colleagues in industry is so critical. They really understand the registration process and what is required, how to engage with the regulators to design the right trial so that these results can be put forward for consideration for licensing. And we got very good feedback. I mean, we talked specifically about the design. They raised some key points, partly relating to biomarkers and risk stratification and thinking about trial endpoints. Now, in fact, for these studies, that endpoint's actually quite easy. We're looking at progression to RA. And it turns out that the definitions we've been using in clinic, you know, the classification criteria for RA actually work very well in this trial setting. And follow-up discussions are ongoing. I'm aware that the American College of Rheumatology have also been in close discussion with the FDA about prevention across multiple rheumatic diseases. So RA, I think we're leading the way there, there's no question. But there are some interesting areas around connective tissue disease and looking at the transition from psoriasis to psoriatic arthritis and looking at the risk of that transition. So things are really motoring. And so I think watch this space. There are a lot of very interesting things coming through the pipeline. But what I would say is that the regulators are really aware of this and understand the benefits of taking this approach.
Of course, last time on mute just to close the webinar. We are just about to finish our allocated time of 45 minutes. So I have a last question for you to close down the session. If I ask you to just describe RTCure in a single sentence, what would that sentence say? I will need a sentence per speaker in the panel.
Well, we want the disease not to occur anymore by having a specific antigen-specific tolerance before the disease occurs.
I think if I were to sum up the RTCure project, it would be that it was a fun and collaborative project that really made great steps in forwards in the prevention of RA.
And I would use that word too, fun. It was a hugely fun consortium with really productive meetings and huge knowledge generation.
Thank you for that. It's a great summary. I would like to just close down the session, thanking you once again, Rowan, Lars, and Andy, for taking the time of being here, talking about RTCure as a closing point for the project, not for the results and sustainability. And remind the audience that if they want to learn more about RTCure, they can still check their website, still alive, with a lot of information about the project. We also have a fact sheet in the IHI site, and we will post this webinar also with more information on the IHI website. If you want to keep up to date with IHI's activity, you can check our website, you can subscribe to our newsletter, or you can follow us on social media. We have LinkedIn, X, and Mastodon, so it's easy to stay tuned to us. With this, we're closing this webinar. Thank you all, and have a nice day.
Thank you.
Thank you, Manuel.
Thank you.
Bye-bye.